Good morning, everyone. I warmly welcome you to the technical session 4 on pest and disease management in agriculture in the 33rd annual sessions of the PGIA. So this session includes 6 presentations in total on the above theme. So to begin the proceedings, let me first introduce you to the chairperson and the members of the judging panel of the sessions. So it is my privilege and I take pleasure in introducing our chairperson, Professor Deepthi Bandara. Professor Bandara has had a long career, 42 years of career as an academic staff member of the Department of Agriculture, Faculty of Agriculture, University of Peradeniya. And now Madam is an emeritus professor of University of Peradeniya. Now, I would like to introduce you to the members of the judging panel who will be evaluating the presentations in this session. So, I would like to welcome Professor Nalika Ranatunga, Department of Agricultural Biology, Faculty of Agriculture, University of Ruhuna, and Dr. Jilushi Damunupula, Senior Lecturer, Department of Botany, Faculty of Science, University of Peradeniya, and Dr. Vindya Basnayaka, Deputy Director, Virus Indexing Center, Department of Agriculture. So with those introductions, I would now like to invite Professor Deepthi Bandara, Madam, over to you to chair the session.
Thank you, Chandrika. Good morning, everybody, and welcome to technical session 4 on pest and disease management in agriculture. A very warm welcome to our 3 judges, Jilushi, Vindya, and Nalika, all of whom I know very well. Okay, so to begin with, I would like to make a few announcements regarding the sessions. The time allocated for each presentation is 12 minutes for the presentation and 3 minutes for discussion. So already you have sent in your presentations and the recorded versions will now be played. And at 12 minutes, you will be signaled that 12 minutes is up. And then there will be 3 minutes for discussion. And at the end of the 3 minutes, 2 bells will be rung to stop the discussion. Yeah, that's the one. So if you have any questions during the discussion time, you can use the raise hand option. And once you are okay with the answers, you can use the lower hand option. So please keep your microphones muted during the presentation. And except for the presenter, the judges, and whoever who wants to ask questions, keep your microphones muted. So with that, I would now like to start the presentations and go to the first one. The title of the presentation is Impacts of Nutrient Management and Crop Rotation on Weed Dynamics in Rice Under Dry Zone, DL1B of Sri Lanka. And the presenters are, or the authors are WMDM Wickramasinghe, DAUD Devasinghe, DIDS Benragama, LDB Suryagoda, and WCP Egodabatta. So I believe WMDM Wickramasinghe will be making the presentation.
Competition from weed is one of main avoided factors cause crop yield losses. Under current restrictions for using synthetic herbicides, controlling weed in agro-ecosystem is challenging. Hence, my research finding on impact of nutrient management and crop rotation on weed dynamic in rice under dry zone of Sri Lanka can be timely important. So let's see background of the research. Weeds, it's not complete or resource. It harbor pest and disease, disrupt water management, and interfere with crop quality. Integrated weed management is widely used to manage weeds in the agro-ecosystem. Nutrient management and crop rotation are most essential component in integrated weed management. So let's see how does nutrient management affect on weed. The weed dynamics can vary according to the type of fertilizer application and their relative nutrient element component, especially nitrogen. So weed abundance can increase with the high rate of nitrogen fertilizer. In regard to integrated nutrient management, it uses blend of organic and synthetic fertilizer. So alteration of soil conditions with organic manure leads to weed suppression. And the slow release of nitrogen is more beneficial to weed than crop time. Let's see how does intensity of crop rotations affect on weed. The crop rotation is most effective long-term weed control strategy. Spatial land resource composition imposed by different crops on weeds, change in management practices, conditions, and time have been reported as effective. But the combined effect of nutrient management and crop rotations on weed dynamics in Sri Lankan rice farming system is scarce. Therefore, we hypothesize that differential nutrient availability with changes of sources and amount of fertilizer inputs and alternating above and below ground crop geometry can exert the pressure on weeds that lead to control them effectively. So, objective of this research was to assess the effectiveness of crop rotation and nutrient management strategy for controlling weeds in lowland rice crop. Therefore, this research study conducted during 4 seasons from 2018 and 2019 March season to 2020 Jala seasons within the farm premises of Faculty of Agriculture, Rajah University of Sri Lanka, belong to TL1B agro-ecological region under irrigated lowland conditions. There were two main factors, namely nutrient management and cropping intensities. Nutrient management included with synthetic nutrient management and integrated nutrient management. It was the main plot factor, and each plot have 3,600 square meter area. When concerned the cropping intensity, it included with rice monocropping and rice maize crop rotation. It was the subplot factor, and each plots have 19 square meter area. In synthetic nutrient management system, 100% of nitrogen, phosphorus, and potassium supply using the synthetic fertilizer. In the integrated nutrient management system, 50% of the nitrogen, phosphorus, and potassium supply using the synthetic fertilizer, and 25% of nitrogen was supply using the organic fertilizer. In here, the phosphorus and potassium with organic fertilizer were not standardized because it was dependent on the quantity of organic manure applied to use the adequate amount of nitrogen with integrated nutrient management system. There were 2 cycles. First cycle included with the 2018 and 2019 March season and 2020 last season, and second cycle included with the 2019 and 2020 March season and 2020 last season. When concerned the treatments, the first level include with the rice monocropping system with synthetic nutrient management, and level 2 defined with the rice maize crop rotation with synthetic nutrient management. When it comes to level 3, it was defined with the rice monocropping system with integrated nutrient management system, and level 4 defined based on the rice maize crop rotation with integrated nutrient management system. Pre-germinated VG300 rice variety were broadcasted with the 120 kilograms per hectare rate, and pre-socked hybrid Pacific 998 variety were planted with 12 kilograms per hectare rate. The weed management in both systems were carried out using the post-emergent herbicides, and the application of inorganic fertilizer were based on the 2013 DO recommendation, and application of organic fertilizer based on the basic dressing of both crops, as well as third crop dressing of the inorganic fertilizer in rice and first crop dressing of the inorganic fertilizer in maize. Let's see what are the parameters observed during the experiment. Total weed density. It measured at the both stages of seedling and heading of the rice crop, and 5-leaf stage and dusting stage of maize crop. Other than that, total biomass and grass sedges broadleaf density and biomass measured at the heading stage of rice crop and dusting stage of maize crop. Data for both crops were collected from the 4 locations of single plot using 0.5 meter by 0.5 quadrant. Firstly, the data were log transformed to maintain the homogeneity and normality of residues. Then, analysis of variance was carried out using the mixed model. In here, nutrient management, crop phase, cropping season, and growth stage considered as the fixed variable, and block was the random variable. The mean was separated using LS means at 5% probability level. Let's see what are the results obtained during the experiment. Total weed density. It was varied significantly with the 3-way interaction of nutrient management by crop phase by season. The maize cropping crop rotation in both synthetic and integrated nutrient management system resulted higher weed density compared to monocropping of rice. Concerning the seasons, the wheat density in monocropping of rice resulted higher wheat density with the March season compared to other season. By the second cycle, the integrated nutrient management system resulted lower wheat density than synthetic nutrient management systems, which resulted lower wheat density with the monocropping of rice. Other than that, the three-way interaction of nutrient management by crop stage by seasons also significantly influenced on the total wheat density. In here, both nutrient management system with all cropping seasons shows declining trend from 20 days after sowing to 60 days after sowing. Apart from that, the three-way interaction of crop phase by growth stage by season also significantly influenced on the total wheat density. The wheat density of maize in crop rotations was greater compared to the monocropping of rice in both stages. And although both crops were cultivated during the 2019 Jala season, there were no any significant differences can identify with the 20 days after sowing during the 2019 and 2020 Maha season. Let's see what happened with the total wheat biomass. It also significantly varied with the three-way interaction of nutrient management by crop phase by season. The wheat biomass in synthetic nutrient management with monocropping in initial March season resulted higher wheat density. However, there were no any significant differences observed during the 2019 and 2000 March season. The wheat biomass increased substantially in the maize crop rotation in Jala season with synthetic nutrient management than integrated nutrient management system. Interestingly, cycle 2 showed significantly lower wheat biomass than cycle 1. Let's look at the variation of wheat types with different treatments. Grasses. Density was higher in the maize crop rotation with synthetic nutrient management, and biomass was higher in the maize crop rotation without any effect of nutrient management. When it comes to sedges density was higher in the maize crop rotation with synthetic nutrient management as in the grasses, and biomass of sedges was lower in the crop rotation in both March season without effect of nutrient management. When concerned the nutrient management effect, synthetic nutrient management resulted higher sedges biomass during both March. However, broad leaves were low in abundance. As previous, both grasses and sedges density and biomass decreased from cycle 1 to 2. Let's look at the discussion point that support the results. The continuous monoculture pattern of rice might have led to higher wheatiness due to the increase of long-term favorable conditions and adaptability, and availability and ability to release mineral nutrient from synthetic fertilizer increase wheat emergence, wheat persistent growth, and wheat dispersions. That is the major reason for increased wheat abundance with the synthetic fertilizer application. Initially, the alternative lowland and upland crop rotation contribute to an increase of wheat density and biomass under both systems because the high interrow space and aerobic conditions within the initial growth stages of maize cultivation. Then the wheat density and biomass suppressed by the system itself due to continuous adaptation of integrated nutrient management system. That happened due to the low mineral nutrient availability with the synthetic fertilizer in integrated nutrient management system, as well as organic manure in integrated nutrient management system can reduce weed infestation. This can happen with the altering soil physical and chemical properties, including water holding capacity, aggregate stability, bulk density, and soil temperature of the organic manure, as well as reducing weed seed bank in soil. And other than that, releasing phytotoxic substance in the immediate environment of wheat seeds also contribute to a lower wheat dynamic during the integrated nutrient management system. Based on these results, we can conclude that continuation of integrated nutrient management system strategies shows its potential ability to suppress wheat density and biomass in the lowland rice-based monoculture system. Even under dry season maize, despite an early high wheat density and biomass in the first cycle, wheat suppression was possible with the rice maize rotation. Hence, the combination of integrated nutrient management with rice maize crop rotation can be possible for decreasing incidence of weeds and their negatives substantially in the rice-based agroecosystem. Those are the reference, acknowledgement. Thank you for your kind attention.
Okay, thank you, Ms. Vikramasinghe. And now the presentation is open for discussion. Yes, Dilshi.
Yes, Ms. Vikramasinghe, it was a nice presentation, and I think it's timely with this current scenario with the fertilizer issues in Sri Lanka.
Yes, ma'am.
So, yeah, I have two questions. One is that you mentioned that organic in your discussion, the latter part, you mentioned that organic manure reduced the weed infestation via the reduction in the weed seed soil bag.
Yes, ma'am.
So can you elaborate on that a little bit? How come the organic manure level decreases that?
When we practice the cultural practices with crop rotations, it's alter the aerobic and anaerobic conditions. So the weed abundance and dynamics are changed with that situation. So that caused to the reduce the weed seeds banks with relevant conditions.
Okay, thank you. And I have, if time permits, I would like to have another question. So with this synthetic nutrient management and integrated nutrient management, you have compared these two systems.
Yes.
So what is the way forward? Like what is the take-home message for the policymakers from your study? Or what is your
sorry,
plan?
madam, I'm not clear. Some interruptions happen with when you say.
Okay.
Can you repeat again, madam?
Yes, sure. So you have compared synthetic nutrient management system versus the integrated nutrient management system, I think, which is timely. And what is your thought on way forward? How can you incorporate your results for the policy making process?
In current situations, the herbicides application is totally banned, as well as chemical applications of fertilizers. So we can recommend those cultural practices and those cultural practices for the reductions of herbicides application. That means we can use those cultural practices for suppress the weeds. When concerned the chemical applications, that means fertilizer applications, we can reduce the chemical applications in half, or we can practice in continuously. That means time to time we can reduce the amount of fertilizer that used in cultivations as well.
Okay, thank you.
Any other question?
Madam, can I ask a question?
Please, Nalika. Yes.
Thank you, Darshika, for the timely and great presentation. I have one question. Since you have been doing it for 2 cycles, I wonder whether the water availability has anything to play with this, because you measured the wheat biomass. So what can you say about the moisture availability in the soil, as well as the availability of, rainwater or irrigation water? Can it have any impact in this observation?
Yes, madam. This is dry zone area, so there are some problems with water availability, as well as sometimes it's there are some heavy rain also. So we need to maintain the proper drainage system, as well as proper irrigation practices when concerned in the weather conditions. So with the crop rotations, maize is the one of major crops that's established in here. It's very sensitive for the water availability, water problems. So we already have experience with those problems with water availability and scarcity, and both situations. That means high level of water and scarcity. So in here we use some extra irrigation practices with some applications with some well water, agricultural well water. With
I think we need to stop now. If you can finish it fast, that's fine. Go ahead.
So I think we use some, we face those both conditions. So we have some good practice to control those conditions.
Okay. So I think we need to end that presentation. Thank you, Darshika.
Thank you.
Okay, so we will now move on to the second presentation in this session, and it is titled Management of Sugarcane Smut Disease Using Triazole Fungicides and Synthetic Elicitors. And the authors are ANWS Tushari and DMD Costa. And the presentation will be done by ANWS Tushari.
I'm Sumita Tushari. I'm going to present our research findings on management of sugarcane smut disease using triazole fungicides and synthetic elicitors. Let's move into the introduction. Sugarcane smut is a fungal disease caused by Sporosodium cetamine. The disease has been reported in all sugarcane growing countries in the world, including Sri Lanka. We can easily identify the disease in the plantation because it produces a long whip-like structure from the apical region of the meristem of the infected cane. We call it as the sorus. Here you can see the sorus with millions of melanized teliospores. In addition to that symptom, there are other symptoms of sugarcane smut disease. After the infection, plants become slender. Here in the middle picture, you can see the slender plants with whip-like sorus, and also leaves may be shorter, smaller, and narrow. In this picture, you can see short and narrow sugarcane leaves with the whip, and also it produces higher number of tillers, ultimately giving a grassy-like appearance to the cane. There are different modes of transmission of this disease. Mainly it is transmitted by infected seed cells because this is a systemic disease. And after formation of the whip, it can be easily transmitted by wind. Also, these tiny teliospores are transmitted by rainwater and irrigated water. Very rarely, it is transmitted by insects like aphids and hoppers. You may think that why we need to manage this disease in the plantation, because smut disease causes significant losses to cane tonnage and juice quality of sugarcane. We have noticed that it causes to reduce purity of juice by 3 to 7%, and also 10% increase of the smut disease in the plantation causes to reduce 6% yield. Or not only cane tonnage and juice quality, it also affects loss of germination. The germination loss is much severe in the ratoon crops. There are different strategies to manage sugarcane smut disease in the plantation. First one is use of resistant varieties. Second one is use hot water treatment. Here you can see this is a huge water tank which is at 54 Celsius. We are dipping the seed jets for 50 minutes before planting in the field. Also, we have recommended to use systemic fungicide and to use healthy seed cane from well-maintained sugarcane nurseries, as well as root out the infected clumps from the fields. Use of resistant varieties is the most effective, economical, and environmental friendly method to control most of the sugarcane diseases, including sugarcane smut. Hot water treatment also useful to control systemic diseases in sugarcane. Program out of infected clumps is also practiced to reduce the inoculum levels in the plantation. However, this is not an economical viable option to Sri Lanka because of high levels of labor cost. Fungicides have been used to manage sugarcane fungal diseases in other countries. Also, experiments have been done on effectiveness of synthetic elicitors like salicylic acid and BTH to reduce colonization of fungal pathogen in sugarcane, especially diseases like red rot. However, no information on the use of salicylic acid or other chemical elicitors to control sugarcane smut diseases in sugarcane growing countries. Therefore, we planned our research with an objective to identify effective fungicides and synthetic elicitors which are having an independent effect on spore germination of the pathogen and to determine the field efficacy to manage sugarcane smut disease. Let's move to materials and methods section. Here I'm going to explain the materials and methods under 3 major subcategories. First activity is evaluation of fungicides and synthetic elicitors under laboratory conditions. For this purpose, we had to prepare a spore suspension. Actually, this spore suspension is a composite spore suspension prepared using vibrative Tilia spores, which were collected from all sugarcane growing areas in Sri Lanka. Simultaneously, water agar media was prepared by amending different concentrations of fungicide and synthetic elicitors. Then we treat this spore suspension on water agar media in triplicates. A negative control was also maintained without adding any chemical. Then the petri dishes were incubated. After the incubation, percentage spore germination was quantified. According to the method described by Bullion et al. In 2012, very briefly, we prepared microscopic slides from each growing cultures. Then 200 spores from each replicate was observed under the microscope versus the percentage germination. Here you can see this is the spore. This is the spore of smut, and this is the germination tube of the smut spores. We observe the formation of the germination tube. The second activity is field evaluation of selected fungicides and synthetic elicitors. As the first step, hot water treatment was done for seed set. Then treatment of fungicide and synthetic elicitors was done separately for this seed set. After that, seed sets were air dried under ambient temperature for an hour. Then the pathogen was inoculated by deep inoculation technique for 30 minutes. After the inoculation of the pathogen, the seed sets were incubated under dark conditions for about 24 hours. Then field establishment was done. There were two varieties comprised in the field experiment, which has different reaction to smut disease. We selected these varieties based on our disease survey data, and also RCT design was used in 3 replicates for the field experiment. The third activity is collection of data, analysis of the collected data. We collected data under 4 parameters. First one is disease incidence, total number of stalks, and number of stalks in smut whips were counted in each replicate from 2 months to 10 months after planting. Average disease incidence was calculated using this formula at the end of the crop cycle. Area under disease progressive curve values were calculated using this formula based on the disease incidence data. As a third parameter, disease severity was determined using this mentioned formula, and percentage reduction of the disease was calculated using the disease incidence data in the treated and untreated plots. A factorial ANOVA was conducted to compare the main effects of independent variables as well as their interactions with the dependent variables. Turkish pairwise comparison was done. The statistical analysis software was used for data analysis. Let's move to results and discussion section. Here I have shown you the results of the in vitro test. Here you can see Tibio has completely inhibited the spore germination from 500 ppm onwards. This is the currently using fungicide, and also hexaconazole. It has completely reduced the spore germination from 250 ppm. While salicylic acid completely inhibits the spore germination at 750 ppm. However, you can see that jasmonic acid or metalaxyl 8% have not completely inhibit the spore germination even at 1000 ppm levels. Here I have shown you the disease variation of disease incidence between resistant and susceptible. You can see that reduction of disease incidence in the susceptible variety has started after 4 months of planting. Both Tebuconazole 500 and hexaconazole 500 have shown zero disease incidence throughout the crop cycle in the susceptible variety, while both treatments of salicylic acid have shown reduction of disease incidence from 7 months after planting. In the resistant variety, all tested fungicide treatments have shown zero disease incidence throughout the crop cycle, and both treatments of salicylic acid have shown reduction of disease incidence after 6 months of planting. That's demonstrated as the late response of salicylic acid for the reduction of disease incidence. Here in this graph, you can compare the variation of susceptible and resistant varieties in disease severity parameter. So, in the disease severity, in the susceptible variety, disease severity reduction has shown after 7 months of planting, and in the resistant variety, the reduction of disease severity has shown 6 months of planting. And also all tested fungicide treatments have shown 0 disease severity in the resistant variety throughout the crop cycle. Here you can see the difference in the area under disease progressive curve values in resistant and susceptible varieties. There's a significant reduction of AUDPC in both varieties except salicylic acid 750 ppm. Here you can see the mean percentage reduction disease for different treatments in both varieties. There's a variation in hexacons of 250 ppm in terms of PRD in both varieties. Other treatments have shown similar reaction between 2 varieties in terms of PRD. Finally, we can conclude that the treatment with fungicides or salicylic acid protects the crop for about 10 months in disease severity and disease incidence, especially the resistant varieties. The following fungicides and synthetic elicitors are recommended to treat seed cane of resistant varieties after usual practice of hot water treatment. These are the recommended fungicide concentrations and synthetic elicitor concentration to manage sugarcane smut disease in our plantations. Thank you very much for your attention.
Thank you, Tushari. So the presentation is open for discussion now. Question?
Can I ask a question, madam?
Please go ahead, Vindhya.
Sumedha, that's congratulations. It's a very nice presentation.
Thank you, madam.
I just want to know, like, instead of using the chemicals, if you use the synthetic elicitors, how practical or how effective will it be?
You mean that instead of fungicide, we have to use synthetic elicitors?
Yes.
Actually, ma'am, we have done that field activities and the laboratory activities, but we have to think of the economical analysis of that one. And also the disease in terms of disease incidence and disease severity, they have shown reduction of the disease in the plantation. So we can use that one for our to manage disease in our plantations.
Okay. No, the reason why I asked is because now there's a chemical ban, you know, right? And you will not be able to find these tebuconazole and hexaconazole and all these in the future. I mean, well, in the near future at least.
Yeah.
So then, I mean, you have done a lot of work and you have come up with something interesting and effective. So if you will not be able to find these, then again, it's just back to square one, you know, you will not be able to. So that's why I was asking whether you can manage without the synthetic chemicals.
Yeah.
Okay, thank you.
Thank you, ma'am.
Okay, any other questions for Tushari?
Can I ask a question, madam? Just a simple one. Yes.
Go ahead, Yulushi.
Yeah, Miss Tushari, you have mentioned that to use as a dip treatment for the seed sets with these, whatever the synthetic fungicides and the synthetic elicitors.
Yeah,
so,
ma'am.
In other countries, are there any other means of using this rather than the dip treatment?
No, ma'am.
For my information, I'm asking. Yeah.
In other countries, because smut is a systemic disease, so in other countries they have using the dip treatment, not any other treatment have been used, ma'am.
So, okay, thank you.
Thank you, ma'am.
Any other questions for Tushari?
Madam,
Nalika, go ahead. Yes.
Short question. Tushari, I just want to know whether, I mean, what is your prediction? Will it be effective for field infection as well? Like you are doing it for the planting material. How about the field situation? Field situation.
Field situations means, ma'am?
Field infection.
In the field?
Yeah.
Yeah. For that, that means, madam, for secondary infections or something?
Yeah, that's right. After establishment, will it protect further, like maybe the systemic ones, ones might affect, but have you ever come across any information related to that?
In our research, we have observed our treatment trial up to 1 year, madam. So in that period, during that period, we could be able to manage the disease up to that. In some treatments, they have not shown 0 disease incidence. So I think in the field, after the treatment, I think we can manage the disease in the field also. That means during that 1 year period, it can be secondary infection in the field also. So in our results, we have shown that it can be managed in the plantation.
Okay, so since the bell has been rung, I think we need to close this presentation. Thank you, Tushari.
Thank you very much, madam.
So we will move on to the third presentation in this session, and the title of this presentation is Prevalence of Bovine Tuberculosis among Cattle and Buffaloes in the Central Province of Sri Lanka. And the authors are YHPSN Kumara, A. Amarasinghe, PGA Pushpakumara, HMS Vasana, WMTD Ratnakumara, GA Gunadana, and B. Alexander. And the presentation will be done by YHPSN Kumara.
Good day, everyone. My name is Sagar Kumara, who is the first author of this study. Today, I'm going to deliver a presentation about one of the most important clinical diseases in dairy industry in Sri Lanka. It is bovine tuberculosis. Before starting the presentation, I would like to take your kind attention to a few pictures which were taken during post-mortem of euthanized cattle. So if you clearly focus on purple arrows of each picture, there were some visible, well-demarcated, different sizes nodules. So those nodules are called as granulomas and are typical lesions of bovine tuberculosis. These samples were later tested for confirmation of bovine tuberculosis using molecular techniques. Therefore, a study about the distribution of bovine tuberculosis is very important to implement preventive measures. The topic of our study was prevalence of bovine tuberculosis among cattle and buffaloes in the central province of Sri Lanka. Bovine tuberculosis is an important leading infectious disease in man and cattle in the world. And this disease is directly associated with people who engage with cattle industry. It's a zoonotic disease that causes respiratory disease in both cattle and humans. So it spreads from cattle to cattle and other wild animals. So therefore, cattle to wild animal borders are at a high risk. Mycobacterium bovis is the organism that causes bovine tuberculosis. It causes huge economic devastation due to culling of animals and loss of productivity. Special treatment for bovine tuberculosis is not recommended as due to its infectious nature. So according to literature, so bovine tuberculosis has not been confirmed as a clinical disease in cattle and buffaloes in Sri Lanka before starting this study. Of course, there were few suspected cases reported in the past. However, those were not confirmed as bovine tuberculosis using molecular diagnosis techniques. There were no proper program established to identify infected cattle and buffalo herds, as also to monitor and control bovine tuberculosis. So therefore, this information will help to develop the present study towards a quantitative estimation of infected cattle and buffaloes in a selected province of Sri Lanka. The general hypothesis of this study was BTB accepted to be present in cattle and buffalo herds in the central province at a higher prevalence. Therefore, 50% of individual prevalence has been set for calculation of the sample size. The main objectives are first to identify prevalence of bovine tuberculosis among cattle and buffalo farms in the central province, and secondly to identify related risk factors for the prevalence of bovine tuberculosis. So this study has been carried out for 3 years, from 2014 to 2017. So medium scale and large scale farms in Nuwara, Kandy, and Madre districts were selected. So both Indicus and both Taurus species were selected as all of them were susceptible for bovine tuberculosis. Sahiwal, Niliravi, and Mura were selected as both Indicus types, and Holstein Friesian, Ayrshire, Jersey, and their crosses were selected as both Taurus types. So this study was a cross-sectional descriptive analytical study, and systematic random sampling has been made to calculate sample size which represent cattle and buffalo population in the central province. The following formula has been used to calculate maximum number of animals which could enhance the accuracy of testing. The farm has been selected as the farm sample unit, and all the eligible animals who were above 6 months were tested for the bovine tuberculosis screening test. According to literature, both Taurus cattle are vulnerable for bovine tuberculosis than both Indicus types, and intensively and semi-intensively managed farms are more susceptible than free-ranging animals, as the disease transmits through aerosols. So therefore, central province consists of many intensively and semi-intensively managed farms where both Taurus cattle are the predominant type. So that was the reason for this population choice. So there were 20 cattle and buffalo farms were selected to this study, and there were 4 large-scale farms and 16 medium-scale farms included. So out of that, many were managed intensively. So the sample size was 616, which consisted of 570 cattle and 46 buffaloes. For this whole study, qualitative tests were used to identify and quantify the infected animals. The first important test is single intradermal comparative cervical tuberculin test, so which was done using Mycobacterium avium and Mycobacterium bovis tuberculin. The picture A shows both tuberculins. The second test was rapid antibody test, which was done using serum of the tuberculin test positive animals. This test actually we have used as an additional complementary test to increase the accuracy of bovine tuberculosis detection. The picture B shows rapid antibody test kit, which was received from Quickening Biotech company. So in selected animals, at first injection sites were marked using an ink. The picture A and B clearly shows how we have done this job. So we have used a pipe and ink to mark Mycobacterium avium site as well as Mycobacterium bovis site. So we have actually used left side of the neck of adult animals, but both left and right side were used in heifers, as they do not have enough room to separate injection site by 15 centimeters. The test positive was decided based on the diameter difference of test reaction sites after 72 hours. So if the bovine reaction is lower than 1 millimeter, the avium reaction, the test animal was considered as negative. If the reaction difference is 1 to 4 millimeters, those animals were considered inconclusive and they were retested after 42 days. So if the difference is more than 4 millimeters, so those animals were considered as positive animals. So rapid antibody test has been carried out using serum samples collected from tuberculin test positive animals. So this test was carried out at a laboratory where temperature was controlled. The picture A shows positive to the test where both control and test bands appeared in red. But picture B shows negative reactions where test band did not show any reactivity. So let's move on to the results and discussion. The pictures A and B shows positive reactions to the avium and bovis tuberculin respectively. So similarly, pictures C and D show negative reactions to the avium and bovis tuberculin respectively. The high reaction to the skin test was detected in many animals as the test sensitivity and specificity is considerably high. So these were recorded as over 70% and 99% for sensitivity and specificity, respectively. So based on the tuberculin test results, the overall prevalence at individual level was recorded as 22.6, and it was 50% at herd level. These figures are considerably high compared to the sum of the developing countries in Asia and African continents. So high prevalence was detected in large-scale, intensively managed old farms compared to the medium-scale, semi-intensively managed new farms, respectively. The number of animals positive to BTB was found at a high rate in Morelia district and then Kandy district. However, 0 cases were detected in farms located in Mathare districts, which was very good in terms of disease distribution. So logistic regression analysis was conducted to identify relationship between bovine tuberculosis and identify risk factors. There were significant relationships were observed between bovine tuberculosis and body condition score, breed, origin, reproductive status. However, age, sex, and parity were not significant. So among those female animals, low body condition score, Aysher and Holston genes, and purchasing animals from another source, and animals that were lactating stage, and also older than 10 years and more than 6 or 5 parities were identified as high-risk groups for bovine tuberculosis. The rapid antibody test was conducted for 33 blood samples, which were collected from tuberculin test-positive animals. However, only 7 were positive to the test. This was obviously because of low antibody titer levels detected in infected animals. Let's move on to the conclusion and recommendations. Tuberculin test is an effective method in bovine tuberculosis surveillance in Sri Lanka, and this test can be performed in all regions of Sri Lanka to detect island-wide prevalence. So high prevalence increases zoonotic potential, and this disease proportionate with scale of operation. PTB is highly influenced by breed, origin, reproductive status, and body condition. It is a zoonotic disease in the dairy industry. So eradication programs must be placed to protect uninfected animals. Eradication is of course expensive as we need to remove infected animals. However, compensation can be done at a government level. Test and sorter policy is the best option what developed countries practice at the moment. According to the literature, segregation on infected animals can be done if the prevalence is very high, but it is not the best option. Animal movement should be restricted, and regular veterinary inspection should be carried out to detect infected animals. So finally, I would like to thank the following people and also University Research Grant for providing funds to conduct this important study. Thank you.
Thank you, Kumara. So the presentation is open for discussion now. Any questions for Kumara?
Madam, I'll ask a question. Is it possible? Yeah.
Please go ahead.
Mr. Kumara, yes, it was a nice presentation, and it's not my field of research, so I'm not very familiar, but I would like to know that you said that you have done this study in the Central Province. No?
Yeah, of course, yeah.
Yeah, Central Province. So are there any studies, because you said that the reports are lacking, so you did, that was the objective of your study. So how about with other provinces? Is it more prevalent compared to Central Province? Any observation done?
Yeah, thanks for the nice question, actually. So this was the initial study that we have carried out in Central Province. So this is the part of my MPhil study. Actually, we have done sort of studies covering all around the Sri Lanka. So we have received random sampling from everywhere in the country, because actually the Central Province is the kind of origin for disease. So what has happened was in the past, so cattle have been distributed among the country. So what happened was the bovine tuberculosis lodged in everywhere. So we received sort of samples from everywhere. We have done bacteriology and pathobiology, and also we have done PCR and sequencing, we confirm actually, yes, bovine tuberculosis everywhere. Yes, that's true.
Yeah, of course. So later on, you will get those results and you can compare with other provinces, no? So
yes, we did actually. So by looking at the kind of prevalence studies, so this test actually, as I mentioned, this is highly sensitive and specific test.
yeah.
So what we can do is like, I think the DAPH, Department of Animal Production and Health, so they have given the instructions to all the veterinary investigation officers established around the country. So sort of investigation running at the moment. So I think so results should be coming up in the near future.
Thank you, Mr. Tushar.
Just a quick question, Kumar, that I had. Your study was done in the central province. I just want to know whether this bovine tuberculosis is influenced by the climate.
Well, of course, I would say like, so it's a very good question. So looking at now, right, I'm in the New Zealand actually. So looking at the past, so this disease spreads, especially in summertime. It's a hot climate, to be honest. Mm-hmm. So because animal movements during the hot climate is very, very, very high. So for example, this is not only in the cattle, so this is also prevalent in the wild animals. So what happened is wild animal cattle movements. So they have a sort of very low barriers. So especially when you go for the Nuwara district. So where we have the distribution is maximum. So that is obviously, I would say climate is possible.
Thank you. Any other questions for Kumara? All right. Okay. So time is up and thank you very much, Kumara, for the very good presentation. Yeah. Okay. Well, so we move on. And the fourth presentation this morning is titled Snap Bean Breeding for Rust Resistant, Validation of Molecular Markers for UR2 Gene Introgression. And the authors are HMPS Kumari, CK Vibad, PCG Bandarnaga, MAP Corrales, and RGAS Rajapaksa. And I guess the presentation will be done by HMPS Kumari.
Snap bean breeding for rust resistant, validation of molecular markers for the UR11 gene integration is my research. In this research, we considered common bean, belong to family Fabaceae species Bacillus vulgaris, cultivated as dry beans or snap beans. Dry beans are mainly for mature seeds, snap beans are for immature green pods. So in Sri Lanka, we are mainly cultivating varieties for immature green pods. So that is belong to snap bean. So it is high value vegetable crop with high nutritional value, and pest and pathogens are among most basic leading causes for yield and quality losses. So rust is among most destructive diseases, and 90 rust races are identified worldwide. So increase of 1% disease severity in bean rust leads to yield loss of approximately 19 kilograms per hectare, reported by many scientists. Bean cultivation in Sri Lanka can be seen like this: grown in upcountry, wet zone and intermediate zone, especially in Badula, Bandar, Nuwara and Kerala region. And temperature, favorable temperature is 20 to 26 centigrade, and average annual cultivation extended about more than 7,000 hectares, and annual production is more than 40,000 metric tons. And in Sri Lanka also, we observe major threats for bean cultivation like virus, rust disease, heat, stress. So common bean rust is the causative agent is Basidiomyces fungus, and species is Uromyces appendiculatus, and it's an obligate parasitic fungus. It has complex life cycle, spread by wind, water, supportive phases, and it's very rapidly spreading disease, and it infects leaves, stems, and plants. So this research activity was carried out, understanding of this problem, because we know it's high income generating vegetable, and this disease is a threat, and it reduces the yield even in Sri Lanka around 18 to 100%. It reduces quality and market value of the product. And disease management strategies, we are mostly using fungicides and cultural practices, but it's very important to develop resistant varieties. Resistant breeding considered the most cost-effective disease management strategy. So single dominant genes control rust resistance. Well-characterized genes are like UR3, UR4, UR5, 6, 7, UR9, 11, 12, 13, and 14. And these are local Bovine varieties, but no rust-resistant varieties reported in Sri Lanka. These are the differential series varieties worldwide identified with genetic background originated from Andean or Middle American gene pool. These are the binary values used to nominate the rust races. So in our study, we considered UR11 gene, and UR11 gene is reported with PI 18-1996 cultivar. So in my research, objectives were like this. Broad objective included validation of DNA markers useful for marker-assisted breeding and developing resistant varieties of snap beans in Sri Lanka. And specific objectives like to validate molecular markers for UR11 gene, to identify resistant new lines with molecular markers, to apply molecular markers for fast breeding. So as methodologies, this research carried out at Horticultural Crops Research and Development Institute, Gangnur, and during 2018 to 2020, Germplasm like PI 18-1996 was used, which carries UR11 gene, resistant to 89 races, and local cultivated variety capital in was used. Popular farm variety, released by Department of Agriculture. And for the screening, rust was collected from local prevalent areas. And new cross combination prepared using recurrent parent and donor parent, selected as like this, compatible nil 18-1996 line. So in this cross, F1, F2, BC1, F1 generations were created, and they were phenotypically and molecular based analysis were done for those populations, and selected resistant lines were used for the development of resistant varieties. And this is the standard rust disease rating scale. This scale included 1 to 6 grading like this, according to the symptom development, according to their severity. So this scale was used to score the disease symptoms. So phenotypic screening carried out with locally reported rust races. Their scores were collected and they were dissolved in tap water containing 20, and they were mixed using stirrer like this, and the spore suspension should be including proper concentration, like 2 times 10 to the power 4 iridospores per milliliter for proper inoculation. So phenotypic screening procedures like this. So we need to plant those population, and at early stage we are selecting those plants for inoculation. We can inoculate leaves using that prepared spore suspension, and we need to keep them in incubator for 18 hours, and then we take out them and keep in glass house until disease development. After 10 to 12 days, we can see disease development like this. In molecular screening, we do DNA extraction using young leaves, using CTAB method, and then PCR and jelly electrophoresis. This is the Pcr cycle, and these are the primers used for this analysis. It included rapid primer and 2 scarf primers, repulsion and coupling phase primers are used. So phenotypically identified plants were used for marker validation. So healthy leaves from those plants were used for DNA extraction, then PCR and lymph analysis were done. These are the results obtained from phenotypic screening. So we could observe immune resistant and susceptible reactions. We did Pcr analysis to get idea about the behavior of that monogenic dominant gene. So we got results as expected, like F2 population shows resistant to susceptible ratio 3:1. We see F1 population so short resistant to susceptible ratio as 1:1. So agrees with our expected values. So in gel images, these are some of the images for different lines. So we got banding patterns as we expected mostly. More than 95% of the results agree with phenotypic screening. So UR11 gene present in PI 18-19-96 has been incorporated into the F2 BC1 F1 population successfully, and scar markers work with them. We could use them to analyze the availability of gene, and those markers will be useful in marker-based breeding, and identification of molecular markers for rust resistance will be also important in gene pyramiding. And phenotypic screening is challenging. You know, it takes about 1 month, mostly more than 1 month. So, but we can overcome that limitation with molecular markers. So, as conclusion, we can say UR11 gene is excellent source for resistant breeding in Sri Lanka, and in cross combination between Capricorn and PI 18-19-96 produce reliable results with molecular markers, and rapid marker and scar markers, even more than 95% of the data agree with phenotypic observation. So higher percentage of selection efficiency was observed with molecular markers. This research is funded by Department of Agriculture, Sri Lanka, Council for the Agriculture Research Policy in Sri Lanka, and Department of Plant, Soil and Microbial Sciences, Michigan State University, USA. And also we got very good support from USDA Maryland Laboratory, providing common bean differential cultivars and technical assistance. These are the references used for this research. Thank you very much for your attention.
Thank you, Kumari. So the presentation is open for discussion now. Any questions for Kumari?
Ms. Kumari, Madam, I'll ask a question.
Sure.
Simple one, yes. Ms. Kumari, thank you for your presentation and with this molecular work you have done. So this with this snap bean, what is the impact in bean cultivation for this with this rust disease? What is the impact for our bean cultivation in Sri Lanka? Is it right?
Yes, madam. In Sri Lanka, we mostly observed more than 18% infection with rust disease, and we observe regular application of fungicides.
Yeah.
So we have to reduce that, and worldwide, most of the scientists using resistant breeding, and then they have successfully identified those genes. So it is very important to incorporate rust resistance with our varieties, because sometimes we observe more about 100% damage with rust in Sri Lanka even.
Yeah. So it's timely that you go for the resistant varieties rather than using these synthetic fungicides. No?
Yeah, yeah.
Okay.
Any other question? Any other question? Kumari, I would just like to know the 3 pictures that you showed.
Yeah.
Are they beans from Sri Lanka or elsewhere?
Sri Lanka.
So are they the usual bonchi?
Yeah, yeah, yeah.
Okay.
Yeah, madam.
Madam, can I ask a question also, please? Kumari, you have done quite a lot of work, and I think it's good somebody starts such work in the department. This is a general question. I'm just interested to know, like you said, we observe even 100% infection, right?
Yeah, yeah.
Disease. Like, where about do we see these? In which parts?
Yeah, madam, in this rust infection, when I did my research, I observed when rust get infection at very early stage to the plant, plant got lost completely. But when rust disease get in later stage, we observe they are surviving sometimes, giving some kind of some amount of yield. But when plants infected at very early stage, in very early to leaf tea leaf stage, they are getting completely dead with our available rust races. So in laboratory test, we observe that condition, but in field level, most of the time, rarely we observed in Badulla Bandarville area.
Okay, thank you.
Okay, right. So time is up, Kumari. Thank you very much for the presentation. And so we move on to the 5th presentation in this session, which is titled Varietal Screen for Susceptible Stage of Saccharum Hybrids for Deltocephalus menoni, Vector of Sugarcane Whiteleaf Disease. The authors are KMG Chanchala, KS Hemchandra, L. Nugaliyadda, VKASM Vanasinghe, and WRG Vitan. So the presentation will be made by KMG Chanchala.
I am Chanchala from Board of Plant Protection. Today, I am here to share my research findings on susceptible stage of Saccharum hybrids for Deltocephalus menoni, the vector of sugarcane whiteleaf disease. Sugarcane is one of the most important plantation crops in Sri Lanka, and it provides livelihood to the several rural communities. The crop is affected by more than 100 of insect pests, and out of them, 18 are homopterans. They cause damages to the crop from germination to the ripening of the crop. Deltocephalus menoni is a hemipteran belong to family Cicadellidae and subfamily Deltocephalinae, which cause severe losses by sucking sap on sugarcane crop and also acting as the vector of sugarcane whiteleaf disease in Sri Lanka by transmitting the disease secondarily. When come to the sugarcane whiteleaf disease is one of the most devastating sugarcane disease caused by phytoplasma, and it can cause 100% cane yield loss due at severe infestation condition. As well, it causes reduction of juice volume and thereby pure obtainable cane sugar. In sugarcane fields, low population of vectors usually recorded in germination to ripening stage fields and higher population of vectors recorded in grand growth stage. Keeping that in mind, let's consider some related factors. Leaf sap feeders usually select plants with higher nutrition, quality, and composition for feeding, and female insects usually identify suitable site for oviposition to maximize development and survival of the offsprings. Also, nutrition composition of the plants varies with the variety and the growth stage. Thereby, the most preferred stage of the plant eventually become most susceptible stage of the crop, and most susceptible cane sugar varieties become most susceptible varieties for the vector, thereby the disease. So, it is a prerequisite to know the susceptible age of the sugarcane plant for the vector for accurate varietal screening program. Therefore, my objectives are determining the most preferred age of the sugarcane plant for optimum feeding of Deltacephalus menoni and determining the varietal effect on the most preferred age of the crop. Here are the materials and methods I have used to achieve my objectives. The research was conducted in Sugarcane Research Institute, Udawalawa, Sri Lanka, and I have used 4 sugarcane varieties with different resistance levels because differences in resistance level can cause differences in the age of susceptibility. The varieties I considered are SL92 5588, SL97 1442, SLC 2009-01, and SL96 128. In here, we considered 6 age categories from 1 month age to 6 months. We have maintained insect cultures to obtain 2-day-old D. menoni adult females as test insects. Here, in studying the relationship between feeding performance and plant age, we considered the amount of secreted honeydew as indicator of plant suitability for feeding. In here, we used parafilm sachet as feeding chambers and water-starved young females introduced to those sachets and attached to the 3rd leaf of the plant. Then they have left 24-hour period on those sachets undisturbed to feeding. After 24-hour period, we have detached the sachets from bottom margin of parafilm sachet, and 2 techniques were used to measure the amount of excreted honeydew. Weighing of honeydew using electronic balance and staining honeydew using bromocresol green treated filter papers. Number of salivary flanges on leaf was counted using erythrocene dye test. Here's the experimental setup. Here in A and B, you can see parafilm sachets attached to the sugarcane leaves. In C, WLD vector in sachet and honeydew. In D, honeydew stained bromocresol green treated filter papers. You can see stains in blue color and stained salivary flanges by erythrocene dye test. Data analysis. In each considered variety, feeding data pulled under 6 categories. Variation of the amounts were calculated using ANOVA, and means are separated by Duncan multiple range test at 5% probability using SAS software. The feeding relationship was studied using the feeding measurement parameters, weight of honeydew, area stained, and salivary flanges. Let's move to the results and discussion. When consider the weight of honeydew, the amount of secreted honeydew was different with the varieties, as you can observe in the figure. feeding increases gradually with the age of the crop. It reached peak at 4-month age, as mentioned, and it reduced gradually after 5 months. Higher amount of feeding at 4-month age was recorded in SLC 2009-01 and SL 96-128. It was low at 4-month age in SL 92. The area stained on bromocresol green treated filter papers. It was kind of similar to the weight of honeydew. The amount of secreted honeydew was different with the variety as mentioned in the figure. Feeding increases gradually with the age of the crop. It reached peak at 4 months and reduced gradually after 5-month age. Highest amount of feeding recorded at 4-month age and highest honeydew production was recorded on SLC 2009-01 followed by SL 96-128. The lowest honeydew production was recorded on SL 92-5588 and SL 97-1442. Significant interaction between crop age and variety on the amount of feeding was recorded during the study. When consider the relationship of weight of honeydew and the area stain, we have observed significant correlation among weight of honeydew and stain area. Honeydew stain areas of different ages showed similar pattern with weight of honeydew excreted by the vector, and significant correlation for each variety was recorded. Correlation coefficient and the equation were specific for sugarcane variety, as mentioned here. In salivary flanges on the leaf, the number of salivary flanges on leaf was in increasing pattern with the age of the plant, and considering the 4 varieties considered, the salivary flanges at 4-month age, the highest number of salivary flanges recorded on SLC 2009-01 followed by SL 92-5588. Then other 2 varieties recorded lowest salivary flanges. Let's move to the conclusions. The trend of honeydew production by feeding is similar on each variety. Susceptibility of sugarcane plant, 40 Menoni, change with the plant age. Sugarcane hybrids are susceptible at 3 to 5 month period for feeding and disease transmission. 4-month age of plants are the most vulnerable for vector for feeding and thereby the disease transmission. 3 to 5 month old sugarcane plants are the best to use in varietal screening against Deltacephalus menoni and increased tendency to have high Deltacephalus menoni population when the crop is at 3 to 5 month age old. Thank you.
Thank you, Chanchala. So the presentation is open for discussion now. Any questions for Chanchala?
Chanchala, I have a very small question for my own curiosity. You know, this honey, I mean, I know this is a standard practice. However, that the honeydew is kind of an excretion of extra sugar that they feed on. Am I right?
Yes, madam.
So depending on the age of the crop, how do you say that the sugar intake that they take like a constant? Is it considered as a constant value, or how do you equalize the intake and the excretion of honeydew with the age of the crop?
Actually, madam, it's standard that amount of feeding is correlated to the amount of excretion. With the age of plant, the excretion amount varies because with age, their ability to suck from those plants vary. Due to some nutrition composition and other physiomorphic characters of the plant leaves. That's it.
Okay, thank you very much.
Chanchala, I have a small question. So according to your results, we saw that the 4th month is the most susceptible, whichever the variety. Okay.
Yes,
and we saw that one of the varieties has a lower infection, or lower excretion than the other 3. Now you also said that the age is related with this particular excretion. How is it that the age is related with the excretion? Because in all 4, the 4th month showed the highest. I mean, the age of the variety, I mean.
Yes, madam. There are peak recorded in 4-month age in each variety. With the age, it was increased, and increasement of the sugarcane plant, it was decreased. That's how their preference vary with that age, because I think it's a nutrition composition of the sap content.
Okay, but mostly my question is, from your results, what is the practical implication? How are you going to use these results to make any intervention?
Actually, madam, if we artificially inoculate the plants with vectors in a wrong age, that can be fails because they do not feed much on that plant to transmit the pathogen to that plant. Therefore, if we only introduce those vectors at susceptible age, we can observe the proper inoculation of the disease and thereby proper reading of the infection.
Okay, right. Thank you, Chanchala. So
can I ask one question?
Please go ahead, Vindhya. Okay, go ahead.
Yeah, Chanchala, this is just for my curiosity as well. I don't know whether I missed somewhere. Into these sachets, how many insects are you introducing? Just one?
No, actually it's 90. 90
per sachet?
For one. Sorry, madam?
Yeah.
Into one sachet, right?
Yes, in sachet one insect.
One insect.
One insect per sachet and 90 replicates.
Ooh.
And 90 replicates.
Yes.
Okay. Is it the standard procedure to introduce only one insect? Because I mean, if something happens to that one insect, then that whole thing is, I mean, we cannot use it, right? I mean, is this the standard?
Actually, madam, usually this kind of studies we use 30, but in case of accuracy, I did 100 and use the undamaged results of undamaged insect sachets.
Ah, right. Okay. Okay. Right. Thank you.
Okay. So I think time is up for Chanchala. Thank you very much for the presentation. And now we move on to the last presentation of this session, which is titled Screening of Finger Millet Germplasm in Sri Lanka for Blast Disease Resistance. The authors are W.M.R. Kumari, D.K.N.G. Pushpakumara, and M.W. Virakon, D.M.J. P. Senanayaka, and H.D. Upadhyaya. So the presentation will be done by W.M.R. Kumari.
Dear ladies and gentlemen, you are warmly welcome for my presentation on screening of finger millet germplasm available in Sri Lanka for blast disease resistance. Finger millet is traditional cereal of many South Asian and African countries. Also, it is staple cereal of some countries in arid and semi-arid tropics. Above all, finger millet renowned as super cereal and nutri-cereal due to its high adaptability, nutritional value, and health properties. In Sri Lanka, it is third important cereal. Presently, finger millet demand exceeds its production and we have to import 3,000 metric tons annually. Considering about the blast disease. Blast disease is a fungal disease caused by Magnifica and it infects wide variety of grasses including rice and millets and have been reported economic damage to these crops. This disease occurs at all the growth stages of finger millet. During seedling, it is called leaf blast. During flowering, called finger blast. And during grain filling stage, infection to panicle stalk cause neck blast. High RH and temperature range from 25 to 38 degrees Celsius favors to vigorous infection of this disease. These are the symptoms of blast disease. Spindle-shaped lesions on leaves, nodes, and heads can be seen. The heavy infection causes seedling mortality and empty panicles. Why this blast disease important in Sri Lanka? Because finger millet grow under low input cropping system under subsistence level. Attention to disease management is very poor and negligible. Further, finger millet cultivation in Maha season, which is the rainy season, having weather conditions are favorable for disease infection and spread. Therefore, host resistance is the most desirable and durable way of disease management. Therefore, having accurate information on blast disease resistant finger millet germplasm available in Sri Lanka is important to develop or direct introduction of blast disease for resistant varieties in Sri Lanka. With that justification, the objective of present study is to identify blast disease resistant finger millet germplasm from some local and exotic finger millet collection available at Plant Genetic Resources Center. A total of 139 local and exotic germplasm collected from PGIA and few farmer cultivars collected from Mahiyangana and Kilimanjaro districts were included in the study. The screening trials were carried out in 3 Maha seasons at Field Crop Research and Development Institute, Mahadupallam. During season 1, high-density nursery beds were established for screening. Therefore, only leaf blast screening was done. These nurseries are normally used to raise finger millet seedlings until transplanted. In order to increase the disease pressure, susceptible check ground are used in borders and every 10th row. The beds were covered by polythene in order to increase the arch. During season 2, comparatively lower density row seeding method was adopted in order to provide adequate spacing for panicle formation. Hence, leaf, finger and neck blast sporing could be done. The plot size was 3 meter by 0.6 meter, single row was per accession was adapted. Season 3 is similar as season 1, high density nursery beds were used. The only difference is one seeding row out of 2 was removed after scoring of leaf blast in order to provide minimum space to panicle formation. Therefore, leaf, finger and neck blast scoring was done. This is the activity plan of pre-season. The artificial incubation was done at the seedling stage. The leaf blast severity index and finger and neck blast percentages were calculated using equation 1 and equation 2. Based on values, the germplasm were grouped into different resistant categories as the information given on the slide. Analysis of variance and rank correlation coefficient were obtained for disease severity index and agronomic traits. Let me describe the results of present study. First, screening of finger millet taxation for leaf blast disease under artificial inoculation. During season 3, leaf blast screening was done in 3 times. At 22 days after sowing, except 14 accessions, all the other accessions were grouped in susceptible category, either moderately susceptible, susceptible, or highly susceptible. But later, 1 week later, all the germplasm were grouped under susceptible categories. Interestingly, 39 days after sowing, all the germplasm were grouped in resistant or moderately resistant category. During season 1, even though high-density nursery beds were adapted, the half of the germplasm accession grouped in susceptible category and the other half was grouped in resistant category. During season 2, the comparatively low planting density was adapted. Almost all the accessions were categorized into moderately resistant category. Let me interpret these results. The gradual decrease of disease severity at 5 to 6 weeks after sowing is due to development of adult plant resistance. Thus, leaf blast severity low at low density row seeding method compared to high density nursery bed due to microclimatic differences. Present study, we could identify higher leaf blast germplasms that showed higher leaf blast severity. They were from cultivars collected from Kilinoche and PGIA collections from Jaffna and Markle. These germplasm are shortage varieties. These are the photographs of season 1 and season 2. Next, we look at the response to finger millet taxation to finger and neck blast under natural infection. The finger blast reactions during season 2, except 11 accessions, all the other accessions grouped the immunoresistant category. Into season 3, all the accessions were grouped under immunoresistant category. Similarly, the neck blast response is, except 2 accessions, all the other accessions were grouped in immunoresistant category. Whereas, into season 3, most of the accessions were grouped in immune category and rest were resistant category. From this response, we could identify 7 accessions and 87 accessions were consistently showed immune reaction for finger and neck blast respectively during both seasons. These are the photographs of immune accessions for finger blast disease. They were all our local collection from Anuradhapura, Matale, Nuwara, Monaragala and Kandy districts. These are the susceptible accessions. The analysis of variants for agronomic traits and disease severities showed that germplasm were significantly different or diverse for agronomic traits and blast disease response. A total of 48 germplasm accessions showed higher yield range 2.5 to 3.6 tons per hectare in the disease screening plots. Even though Ravana used as a susceptible check, it gave the higher yield compared to all the other accessions. Majority of germplasm were flowered greater than 27 days after sowing. These are the photographs of some high yielding accessions. Finally, let me present the correlation between agronomic traits and blast disease severity. The days to flowering was significantly and negatively correlated with finger and neck blast, while days to flowering was positively correlated with grain yield. However, leaf blast does not show any significant correlation with other agronomic traits and leaf and finger blast during both seasons. Finger blast is positively correlate with the neck blast, and neck blast is negatively and significantly correlate with gray meal. It is obvious because neck blast falls to empty panicle and it directly influences the gene. Finally, as a conclusion, from the study, germplasm showed diverse response to blast disease as well as yield and flowering duration. However, germplasm rarely shows the leaf blast resistance at seedling stage. But from this study, we could identify highly susceptible germplasm for blast disease. Further, we could identify consistently immune reactions, consistently immune reactions to finger and neck blast. Therefore, these results can be used to develop or direct introduction of finger millet blast disease resistant finger millet varieties in Sri Lanka. So, finally, finger millet is believed to be a certain crop for uncertain future. Before I wind up my presentation, let me acknowledge Apari for financial support and all the directors of Mahailu Pallama and research and field staff of the Mahailu Pallama. Thank you very much, ladies and gentlemen, for your kind attention.
Thank you, Kumari. So the presentation is open for discussion now.
Kumari, I have a small question. At the moment, what is the most widespread accession in Sri Lanka? I mean, for the cultivation.
Actually, we have released two varieties. Department of Agriculture have released two varieties, Ravana and Oshada. But when I survey about the varieties, farmer varieties, there are a lot of farmer varieties grown by farmers in different districts. The morphological similar to Oshada during my morphological studies. Actually, there are a lot of farmer varieties apart from the department varieties. That's all.
So you mean that they are susceptible to the disease? Because I have seen blast in many of the cultivations that we have down here. So like, is it wider spread among all the accessions, like normally cultivated at the moment?
From this screening study, what I was found from these 139 accessions, almost all the accessions were susceptible for leaf blast disease at seedling stage. It's very rare to find a resistant variety for leaf blast. But therefore, because of our general practice by the farmers is broadcasting method, microclimatic conditions and the broadcasting method is high density and cause to the spread of this disease and seedling mortality.
Okay, thank you.
Any other questions for Kumari? So is finger millet grown in all agroecological regions in the country, Kumari?
Not in all, but in dry and intermediate, up country and low country as well. Not in the wet zone.
Okay.
I have, sorry, Vedas, can I?
Yeah, please go ahead.
A small question, like what is the correlation between the flowering duration and the infection of the fingers, like the finger blast? Is there a like correlation?
Yes, the shortage varieties showed highly susceptible for leaf, finger and neck blast from this present study.
Okay, thank you.
Okay, so that actually brings us to the close of this presentation, and time is up. So thank you very much, Kumari. I think with that, we finish this session, technical session 4 on pest and disease management in agriculture. So I take this opportunity to thank all the authors of the papers presented today. Thank you very much for your hard work, and to the supervisors who have been supervising these students, and to the presenter, the main author of the paper. Good luck and congratulations on future work. And then I would also like to thank our 3 judges, Professor Nalika Ranatunga from the Department of Agriculture Biology in the Faculty of Agriculture, University of Ruhuna, one of my best students, and Dr. Vindya Basnayaka, Deputy Director Research at Plant Virus Indexing Center at Homagama, and Dr. Jilushi Damanpula, Senior Lecturer at the Department of Botany, Faculty of Science, University of Peradeniya. Thank you very much, ladies, for your cooperation and assistance in judging this session in the 33rd Annual Congress of the PGIA. Thank you very much for supporting our students and for making available your very valuable time. And so now I would like to close this session with thanks to Dr. Chandrika Pereira, my colleague in the department, who has been coordinating this session, and also to my young colleague, Ms. Virajini Bulat Sinhala, for keeping time so accurately during this session. So thank you very much, everybody, and good luck to all of you in the future.
Okay, thank you very much, madam. Now, stay connected with us. There are some information from the Congress office. So, but before we move on to them, I would like to thank Madam Dipti Bandara for sharing this session. And there is a token of appreciation awarded by the Congress Office of PGIA for your kind cooperation in this.
So they are trying to give me some souvenir. Thank you, Virajini. I did not expect this, but thank you very much. So that was very nice of them. So is that all then, Chandrika?
Yes, madam, there is some more. I would also like to thank the panel of judges and would like to request of them to stay until until we confirm the submission of marks. So, and then the participants, we have the invited presentation of Professor Deanne Beckel from the University of California, Davis at 1:30. So the organizers extend you an invitation to attend this session at 1:30 through the Zoom links that are provided to the main session. So you can join this session at 1:30 using the given links. Yeah, the links are there in the PGIA website, and also you can log in via the Facebook and the YouTube channel that is given. And now there is another exciting presentation in line for you, that is the PGIA as the pioneer in postgraduate education in Sri Lanka offers 31 different disciplines, master's programs in 31 different disciplines, and through 11 boards of study. And